All right, we're almost there with getting through all these different types of inheritance patterns. So we have one more that's kind of interesting, and it's called codominance. So sometimes in a population there is more than two alleles that are present, and sometimes two of those alleles are both expressed in an individual. And that's what we call codominance. So an example of this that everybody's probably familiar with is your blood type. So you're probably familiar with the fact that in humans there's blood type A, there's blood type B, there's AB or O. And we're not going to talk about the positive or negative because that's something different. That's your Rh factor. So if you have blood type A, that refers to the fact that you're producing what we call an A factor. And this A factor appears within the membrane of your red blood cells. So here they're designated it as these little yellow triangles that are on the outside of the red blood cell. If you have B blood, then on your red blood cells you have the B factor represented by these little circles. If you have AB blood, guess what? You have both the yellow triangles and the yellow circles. So in other words, you're making both the A factor and the B factor on there. So notice, in this case, the genotype of the individual, we're using the letter I, and we're superscripting A and B to represent these A and B factors. So we're using a capital letter for both the A and the B because they're both dominant. They're both being expressed. It's not incomplete dominance. We're not seeing a third phenotype. It's not like we're mixing A and B and we come up with C. Okay? Both A and B are present, so it's called codominance. Now, if you have blood type O, your genotype is two recessive I's. So in other words, you are not producing anything on the outside of your red blood cells. There's no factors at all. So if we go back to our A blood type, which is the phenotype, there are two genotypes to go along with that. They could be homozygous dominant, or they could be heterozygous, where they have that kind of recessive I for producing no factor at all. Same thing with B. You could be homozygous for the B factor, or you could be heterozygous and have that recessive I. All right. So codominance refers to the fact that if you are AB, you're producing both A factor and B factor. So let's look at a problem. All right, let's see if we can solve a problem that deals with this. Okay, so if a mother with type A blood, so if she has type A blood, we're going to use the capital letter I. We know she has at least one allele that's making an A factor, and the other one we don't know about yet. So if a mother with blood type A has a child with blood type B, all right, so I'm going to draw an arrow going down. This is her child. Her child has at least one B allele because he's got blood type B. We don't know what the other allele could be. So then it says, what are the possible genotypes of the child's father? So we want to know what two alleles the father carries. So this B allele here in the child, did they get the B allele from mom? If that was the case, mom would have a B allele here, right? If I write that in. But now mom is now AB. She has AB blood and not A blood. So that can't be correct. So we know that the B did not come from the mom. The B had to come from dad. Okay? So dad has a B allele. So what are the possible genotypes of the child's father? So it's asking for the genotype, not the phenotype. So we know dad can have a capital B. Can he have two capital B's? I don't see why not, right? Either way, he would be passing that on to the child. What's another possibility? Could dad be... He's got B blood, right? We got to pass on the B. But what if he has the recessive for producing no factor. He could still pass on this B to the child, and that would work. Could Dad be AB? So if Dad was AB, right, he would have an A allele, and he would have a B allele. There's the B. So he could still pass on that B allele to the child, so AB would work. Or what if he was O? Would that work? If he was O, he would have two recessive alleles. So that wouldn't work, even though we don't know what his other allele is for B blood, but he's got to get that B from the dad because he's not getting it from the mom at all, right? So I is not going to work. Oh, here's one other thing. AB is not going to work either, right? I just reread that and it says the blood type. No, it doesn't. Okay, sorry about that. The blood of the child is B. All right, it doesn't say anything about the dad. So dad could still be AB. Okay, so dad could be homozygous, or dad could be heterozygous for B, or dad could be AB, right? Those are our possibilities for the child's dad. All right, let's look at the second problem. So it says, Henry believes he is the father of Ryan. Ryan has the blood type AB. All right, so let's write out Ryan's blood type. He has A blood, A factor, and he's got B factor because it's codominant. So that's his genotype. That's Ryan. Ryan has blood type AB. Henry has blood type O. All right, so Henry has two recessive alleles. Can Henry be Ryan's father? Well, considering one of these alleles has to be given to Henry, or excuse me, to Ryan, all right, then it doesn't look like it's possible, right? So Ryan inherited an A from one parent. He inherited the B from his other parent. If Henry has the recessive for no factor, there's no way that he could be Ryan's father, all right? So Henry is not Ryan's dad, sad to say. All right, one other thing that we want to talk about is what's called nondisjunction. All right, so we've been talking about inheritance and how alleles are kind of segregated out into the respective daughter cells. We talked about this with the law of independent assortment, right? So sometimes what happens is chromosomes don't always separate out perfectly. And this can lead to what we call trisomies and monosomies. So you may have heard of trisomy 21, which is Down syndrome. And so this can be caused by a process that's called nondisjunction. So what happens if we look in this first image here? So we have a cell and we have a set of small chromosomes, homologous chromosomes, and we have a set of large homologous chromosomes. And typically sister chromatids are reaching out and making connections from either side. And during anaphase, homologous chromosomes would be pulled apart. But what happens if one of these spindle fibers does not connect to our homologous pair? So the spindle fiber on this side will pull both members, right? Both members of homologous pair are going to go off to the left and none will go off to the right. And that's what you see after meiosis 1. The result is that we've got extra chromosomes in the daughter cells here of the large chromosome, and this one's missing the large chromosome. As we go through meiosis 2, they get separated out further, right? The sister chromatids get pulled out into separate daughter cells. And so now we have 2 copies of the large chromosome and just one of the small, which is as it should be. in these two daughter cells, there's no large chromosome at all. So if you think about it, what would happen if, let's say we had a sperm cell here and it, let's make it red, had a large chromosome and a small chromosome, which should be the normal complement, right? So make that large, or I'm not making it larger, I'm just making it wider. So we had one large, one small chromosome. So if this one was fertilized by this sperm, then we would have 3 copies of the large chromosome, right? So there's 1, 2 there, and then it would get the third one from the sperm. And then it would have 1 copy of the small chromosome and 1 copy of the small chromosome from the sperm. So now we see 3 copies of the large. So this is where we could have 3 copies of chromosome 21 because one of the gametes carried an extra copy. Now, what if instead of that gamete, this one was fertilized by that sperm? Then we would have the 1 large chromosome from the sperm, and we would have the 2 small ones. Okay, so now the individual is missing the other large chromosome, and this is what we would call a monosomy because they only have 1 chromosome from one type of that chromosome, or just 1 chromosome, I should say. Okay. The other side here is showing you how nondisjunction can occur not at meiosis 1, right? So meiosis 1, let's say, happens as normal. Homologous pairs are pulled into the 2 daughter cells that are formed. But now it's showing what happens if sister chromatids are not pulled to opposite poles. So let's say the spindle fiber is not attached on the right-hand side here, and sister chromatids end up in the same daughter cell. So now we have 2 copies of the large chromosome. In the next cell, there's no copies of the large chromosome. Over here, though, we have 2 regular daughter cells that are produced, and they'll have 1 large copy, 1 small, 1 large, 1 small. So this is how it normally should be. And this is where nondisjunction has happened, causing additional chromosomes. So this one, if it was fertilized, would cause a trisomy, and if this one was fertilized, it would cause a monosomy. Okay, so this is what we call nondisjunction, when the chromosomes are not separated during meiosis in the production of gametes.